Hello, biologists. This is lecture number 7 on mitosis. This is the last of the information that will appear on midterm number 1. So get ready, here we go. In this lecture, we're going to be talking about cell replication, cell reproduction, and there are many reasons why your cells would reproduce. Think about this. When you are a single-celled organism right after conception, that process goes from a single cell to eventually the trillions of cells that you are made of, and heck, that's mitosis. That's what's going on here in this little girl as she approaches adulthood. This lizard right here has broken off its tail. Well, how does it replace its tail? Through mitosis, through cell division. Here's another great example. How about this? This is my buddy that was really smart and tried to snowboard off the back of his truck, and that's kind of what ended up happening to him. So what's going on there? Well, he's bleeding. But there's more going on than just that. It turns out that his skin cells are reproducing as we speak, and they will eventually replicate and cover the hole and heal himself. Well, where does all the information for how to do this come from? Well, it's all right here in your DNA. So your DNA, those are the molecules that are in virtually every cell of your body with all of the genetic instructions for how to make you. DNA stands for deoxyribonucleic acid. We implied in the macromolecule lecture that actually this is a type of nucleic acid. So a pop quiz, how many of you remember what components make up a nucleic acid? Let's see, you have a sugar component, you have a base component, and you have a phosphate component. If you have those three things, you have a nucleic acid. DNA is one such molecule. It's a very, very long molecule, though if you were to actually stretch this thing out, it would be 2 meters long. That's really, really long. That's like almost 7 feet long. So that's contained in one cell, and it goes through a series of coils. So here's kind of like a visual representation of what DNA looks like. Of course, this is kind of stylized. But it goes down, it gets wrapped around a whole bunch of little proteins, and then wrapped and coiled some more, like this. And then these coils get coiled until eventually they can actually condense down into what are called chromosomes. And chromosomes are interesting. Chromosomes are a single strand of DNA, and in the human body we actually have multiple strands. That is, we have multiple chromosomes, and the material they're made up of is just loosely called chromatin. Chromatin is all of the DNA material that happens to reside within a cell, within a nucleus of a cell. All organisms contain a different number of chromosomes. These are just some kind of classic examples here. Asparagus. Okay, so asparagus actually has 30 chromosomes, and catfish have 58 chromosomes compared to fruit flies, which only have 8 chromosomes. Now, these chromosomes contain the DNA instructions on how to make the entire organism. Everything from the scaffolding of how the body gets put together, to the biochemistry that goes on in them, to the physiology, to actually behavior. In the case of organisms, a lot of the behavior that you exhibit, it's contained in your DNA too. Okay, notice these numbers here are all even. That's because the chromosomes that you contain in your body, you actually have 2 pairs of each. Okay, what does that mean? Well, I like to think of chromosomes and DNA as being an encyclopedia. You guys remember what an encyclopedia is? You remember you used to actually go and see that at school, where they'd have a whole series of books on all kinds of topics starting from A to Z, and there was so much material that the A volume actually was a book unto itself, and then the B volume was a book unto itself, all the way along. And so you got your encyclopedia of how to make you from your mom and from your dad. You got one copy from your mother, you got another copy from your father. Using this encyclopedia analogy, it's kind of like saying the A volume in my encyclopedia, starting from A to Z, the A volume, I have one copy of that A volume from my mom, and I have one of the A copy volumes from my father. And so it's kind of redundant. Your DNA system's redundant. You have 2 copies of information. So when we show you these here, and you notice they're all even numbers, that's because there are 30 chromosomes of asparagus, but really there's 15 pairs. They've got 15 sets of instructions. They just have it doubled. Okay? And notice gorillas, one of our close relatives, they've got 48 chromosomes. Okay, so that's chromosomes. But let's talk about how cells divide. How does one cell lead to two cells? Well, obviously, the genetic information that is contained in your cells has to duplicate itself. It has to photocopy itself before the cell can split. Because when you're done with two cells, they have to have the same information in them, the same DNA. So, cell division actually involves a multitude of steps. The dividing up of the DNA is that first step. And this is kind of what it looks like. What you're looking at here in this micrograph, this picture right here, is actually of what are known as sister chromatids. So let's imagine that we have chromosomes in our body, but we're only going to focus on one. This is what the chromosome looks like, let's say. Prior to the actual cellular division, before the cell even splits, the DNA has to duplicate itself. It has to copy itself. So it's literally like photocopying the information. And this happens in every chromosome. What is being shown here is just one, for example. So after it duplicates, each side here is identical to the other in mitosis. So this sister chromatid on one side is identical to this sister chromatid over here. After cellular division, that is, now that the DNA is duplicated and after the cell has actually split and divided, now you end up with the identical copy in either side of the chromosome. So it's the same as what started out up here. Down here, it's just been duplicated. So it makes sense that going from here to here, you have to duplicate the DNA in the process. Make sense? Okay, so you actually get one copy of DNA from your mom and one copy of DNA from your father. So here's your dad DNA. This is, again, to keep our encyclopedia analogy going, this would be like my A encyclopedia chromosome from dad. This is my A encyclopedia chromosome from my mom. And now we showed you all of those chromosome counts for those other organisms, but in humans, you have 23 pair, or a total of 46. That is to say, you have 23 chromosomes that came from your mom, and you have 23 chromosomes that came from your dad, and they are more or less redundant. We're going to cover how that works a little bit later as far as what are the ramifications of having two copies, two sets of each chromosome. But we'll deal with that later. Right now, just accept that we have 46 chromosomes in your body, 23 pairs, and just prior to cellular division, these chromosomes divide, or they multiply themselves. And so here, chromosome encyclopedia A from mom has duplicated, and chromosome encyclopedia A from your dad has duplicated. And again, this happens with all the chromosomes, not just these ones, obviously. All 23 pairs of them. So mitosis, and so here you have your identical sister chromatids. Okay. Mitosis is really all about nuclear division, and it maintains the parental chromosome number in those two cells that end at the end. We call them daughter cells. So you have a parent cell that leads to two daughter cells once it splits, but in the process you have to make sure that the DNA that you started with is the same DNA that you ended with. All you've done is Xerox copied them. You just photocopied them so that there could be an identical copy in the two daughter cells that came about. And the process of how this happens is actually quite straightforward, which is why this lecture is going to be a whole lot shorter than some of the others. And so the whole process is called the cell cycle. All cells kind of go through this, and it's a sequence of events that happen between the mitotic cellular divisions. And so what do you start with? Well, we're starting with a new cell. Imagine that we're starting with a new cell. And over time, that cell is going to increase in size, increase in mass, and eventually it's going to duplicate its DNA. After it's duplicated its DNA, then the cell divides. And, again, this is a very straightforward process, and I've got a couple of videos up that I want you to check out. They will talk you through the process. I'm going to walk you through it just right now, just to help you kind of understand this, but you're going to want to look at these videos repeatedly over time. So let's go ahead and start with the cell cycle phases, the different phases that occur during this cell division process. The first phase is called interphase. interphase. It is basically the time when the cell is basically just doing its everyday thing before it's actually ready to divide. Now, that's this section over here. Notice that says interphase. It's a huge portion of the cell cycle. And during this phase is when the cell is going to be growing, when the cell is eventually going to be dividing its DNA up, then it's going to continue growing until it's ready to undergo the next stage, which is mitosis. Mitosis, in this little thin sliver here, is where the nucleus actually divides. The nucleus within the cell divides. And during cytokinesis, that is where the cellular contents, all of that cytoplasm stuff on the inside, also divides, and then the thing splits up, and now you have two cells. So again, you've got one cell. That one cell goes through interphase where it's growing, it's growing, it's growing. Its DNA splits, it grows a little bit more, and then it goes through mitosis and cytokinesis to break it into two cells. So interphase, again, is this longest part of the cell cycle. It's this huge section here. And during this, and by the way, this is what it looks like. If you were to look at cells like what we do in the Biology 109 lab, you actually get to see this during the mitosis lab. But yeah, this is what it looks like. It just looks like a cell with a nucleus in it. Nothing really all that special going on in there, or at least so we see. And here are the various phases within interphase. We've got a G1 phase, an S phase, and a G2 phase. During G1, this is right after the first division happens, so the cells have already divided. You're starting off with a fresh new cell now. And the cell is growing and growing and growing. It's taking on more nutrients, it's increasing in mass and in its volume, and then it hits the S phase. The S phase, S stands for synthesis, or I should say G stands for growth. So S stands for synthesis, and this is the point where the DNA actually replicates, where it turns into those sister chromatids. So long before the cell even goes through mitosis, way back over here, is where the DNA has duplicated. Then after the duplication has occurred, G2 comes along, and this is where you're into like the second growth phase, getting ready for cellular division, which happens next. Okay. So let's go ahead and back up to that S phase and remember what is going on here. That S phase is where the chromosomes are actually duplicating themselves. So we start off with a chromosome here, and remember, you've got 23 pairs, right? We're just looking at one right here. This one duplicates itself through a process we're going to learn a little bit later, and so right here it's single chromosome right here, single chromatid, that then duplicates itself into sister chromatids. Then it undergoes mitosis, and that's when it actually splits. So if you wanted to look at it another way, it looks kind of like this. Here is your pair of homologous DNA. Let's just pretend that these are chromosome number 1. This is chromosome number 1 from your mom. This is chromosome number 1 from your dad. And because these things are kind of contain similar information in them, like hair color or eye color or how tall you're going to be, that sort of thing is contained in the DNA. These are considered homologous pairs. Homologous. Homologs means that they are similar. They are both encyclopedia A's, for example. They're both chromosome number 1s. In your body. Now, does that mean that these DNA strands here are identical? No, not necessarily. Think about what color your mom's eyes are and what color your dad's eyes are. They might be the same, but some of you may actually have parents with different eye color, right? Maybe your mom has blue eyes, your dad has brown eyes. Well, the place on your DNA that actually codes for eyes, that actually determines what your eye color is going to be, it's contained in the same location on these strands of DNA, but this one over here from your mom may say, make brown eyes. And then over here from your dad, it might say, make blue eyes, right? So that's why they're homologous. They're not identical. They're just homologous, okay? It's kind of like having two different copies of a cookbook. And I'm going to turn to the same page in both cookbooks. And yes, mashed potatoes is on both pages, but the instructions on how to make the same thing is slightly different. Okay? You might make one with mixing a little milk in it, the other one maybe you don't have any. So those would be homologous. These two things are not identical, they are homologous. During the S1 phase, though, that's where these two DNA strands duplicate themselves. They have to duplicate themselves in anticipation of eventually going through mitosis, where they will get split. Okay, so let's go ahead and go on to mitosis. This is after the G1, the S, and the G2 phase. So we have already now gone through that growth period. Now we're ready to actually begin to divide up the nucleus and then eventually divide up the entire cell. Okay, mitosis, that very rapid but extremely interesting section actually has 4 stages that occur during mitosis. So the 4 stages are known as prophase, metaphase, anaphase, and telophase. Now, let's go ahead and back up a little bit so we can work our way into mitosis. Let's assume we've already gone through G1, S phase, and now here we have G2. Okay, the cell is ready to divide. This first section that comes from G2, when we enter the actual mitosis, is prophase. During prophase, we've got, you'll notice in here that the nucleus, all of the DNA in the nucleus begins to coalesce. It begins to granulate. And then all this stuff begins to line up during the next phase, metaphase. It begins to line up in the middle of the cell, and then during anaphase, the DNA that was duplicated way back in the S phase then begins to split. It begins to separate apart. And during telophase, well, now they're actually being pulled apart. And then I've actually thrown in the last stage here. Cytokinesis happens after mitosis. Once the cell nucleus has been successfully divided, then the cell itself begins to divide. Okay, so let's visit these phases in a little bit more detail. We're starting off with prophase here. Oh, and by the way, go check out the mitosis video. I've got a whole bunch of really nice videos up here to help keep this straight in your mind. Prophase. During prophase, this is when the nuclear membrane actually breaks down. Let's picture this as a cell here, and here's the nucleus with all the DNA in it. During prophase, the nuclear membrane begins to dissolve and disintegrate, break down. And all of those duplicated chromosomes begin to condense down. At this point, they are no longer 2 meters long, right? They've condensed down to being microscopic in size, but really, really dense, where you can actually see them under the microscope. Okay, they look kind of like this over here. Now, the spindle fibers in the cells, spindle fibers actually begin forming in the cytoplasm. And you use the spindle fibers to move the chromosomes along. So once this nuclear membrane has been successfully divided, then the spindle fibers can then reach in and start grabbing onto that DNA. And the nuclear envelope is now completely broken up at the end of prophase, entering into metaphase. During metaphase, those microtubules have reached out, grabbed onto the DNA, and pushed and pulled them so that they are now aligned right here on something we call the metaphase plate. You could think of it like the equator of the cell. Now, the kinetochore microtubules actually attach to these sister chromatids. Remember, see these DNA fragments? They're still attached. These chromatids are still attached to their sister chromatid. And the centromere region, which is this central nodule that is present in the center or close to the center of each of these chromosomes, is where these things attach. And as they push and pull, these chromosomes begin to line up on that equator. Kind of neat. And then they begin to pull. And as they pull, the cell actually starts elongating. But once they've detached, once these sister chromatids have now been detached from each other, the resulting chromatids that have now been separated begin to get pulled towards the poles. Okay? And once they're separated, each chromatid is now considered its own separate chromosome. Okay? Now, pull them all the way to each end of the cell, and you are now into telophase. during telophase, the chromosomes begin to decondense and go back to kind of unraveling a little bit. And you now have the nuclear membrane begins to reform around the two new groups of chromosomes, the two sets of chromosomes. And at this point is when a cleavage furrow begins to form right in the center of the cell to begin to divide up the cytoplasmic material. Okay? So the results of mitosis actually look like this. You end up with two daughter nuclei, all right? And each has the same chromosome number as the parent cell. So remember, the parent cell started out with 23 pairs if it was a human. In the end, you have two cells, and each cell also now has 23 pairs of chromosomes. And the nuclei are now divided, but you still have one step to go, right? We still have cytokinesis. You still have to divide up all of the mitochondrion and the lysosomes and all of the other things that are floating about in the cytoplasm of the cell. So that's what cytokinesis is. Most cells actually do cytokinesis. You can think of it like skin cells are doing cytokinesis. However, not all cells do. There are certain cells like your skeletal muscles, your muscle cells, that don't do this. Now, something that's really important is that I know that you all probably have the correct textbook, but if for some reason you manage to get your hands on an older textbook, just realize that those old textbooks combine cytokinesis with telophase, and we're not going to do that. Anyway, this is a nice kind of view of how it looks. You have your two cells here that have already gone through mitosis, that is, their nuclei are already divided. The DNA are already contained in two new nuclei, but now we're going to be dividing the cellular material, all of the stuff on the inside. So here is a cell that is or two cells that are essentially almost done doing cytokinesis. Okay, so cell division. Cell division is determined genetically. What do I mean by that? I mean that some cells divide much more quickly than others. Your skin cells are dividing rapidly on a daily basis. Your nerve cells, however, are not. Red blood cells are constantly dividing, but liver cells don't divide nearly as frequently as your blood cells. So some cells divide constantly, others divide rarely, and then so here's a good example, as I mentioned, of a couple of cell types that do it rapidly: bone marrow, making red blood cells, and skin cells. But, and so, go and check out the mitosis in situ video. This is an incredibly cool video. It's only like 30 seconds long or something like that. But in situ means like actually in a test tube with live cells. You can actually observe these cells dividing right before your very eyes. It's actually quite amazing. Okay, so we've already talked about the red blood cells. Now, what isn't a good example of a cell type that does not divide, or at least very rarely? Well, the one I'm showing you here. Nerve cells. They don't divide. Spinal cord injuries become really bad because of this. Nerve degeneration diseases are really horrible because of this. These cells just don't divide after they have already been formed during embryonic development. So having said that, we have now been able to unlock the amorphous stem cells, if you've ever heard of stem cells, and figured out how to revert them back before nerve cells have been basically predetermined. You can start out with stem cells and actually make new nerve cells. So what type of research looks promising for repairing nerves? Stem cell research. Go check out the stem cell video that I have up online. It is proving to be one of the most amazing medical applications in the history of medical science, is unlocking stem cells and being able to get them revert back to that embryonic state where you can actually turn them into any cell you want. Okay, can you guys think of a disease where there is a problem with too much division? If you said cancer, you'd be right. Cancer is really all about uncontrollable cell growth. So, mitosis, we're talking about cell division here, cells making more cells. Well, that's great when it's happening as prescribed by your DNA. But if for some reason you have some problem where the cells don't stop dividing when they're supposed to, that's when you get cancer. And so how can understanding cell reproduction help find a cure? Well, obviously, if we understood the mechanism by which, or I should say the chemical cues that help us understand how cells signal when it's time to divide and when it's time to stop dividing, well then we could probably cure a lot of different kinds of cancers. Okay, so this is uncontrolled cell growth. Now, how many times can your cells just replicate themselves over and over again? I mean, the skin cells that you have now, are they basically descendant of the skin cells that you started out with when you were a baby? Well, yes. So what happens? Why can't they just keep going forever? Well, because eventually they wear themselves out. So what does it really mean to get old? Your cells cannot divide indefinitely. And when they stop dividing, that is essentially when the organism begins to die. And the number of actual divisions that your cells can do and the rate of divisions is actually under genetic control. That is to say that people who tend to live a really long time, like into their hundreds, it is written into their DNA that their cells are actually able to divide more than people who tend to die in their 60s, for example. So anybody know who this person is here in this picture? Anybody? So you older students probably recognize her. That's Raquel Welch. And Raquel Welch was born September 5th, 1940. That's right, she is 71 years old. My goodness, 71 years old, and she looks outstanding. So this is an example of somebody whose DNA it has information on how to continue to divide well into your 70s and still have lots of elasticity and still look great, still be very attractive. But there's the opposite case as well. There are those who have a genetic disorder called progeria. And in this case, cells cannot divide for very long, and they only divide into about by the time these people are in their late teens, mid to late teens. These individuals live very short lives. They usually die between the ages of about 10 to 15. So make sure that you go and check out the section in your textbook on cell cycle regulation. Whether you live a very long life or whether you live a very short life is usually determined by cell cycle regulation issues. We're not going to actually cover it here in this lecture, but I do urge you to go and check that out in your textbook. And that ends the mitosis lecture. I will see you all in cyberspace.